So who's bold enough to start off? I have a question maybe, Sam, you can't answer. But we're talking about connected health, and we heard a very fabulous, not story, but actual things that are going on with epilepsy. And so when you listen to him, do you see some role how CRISPR could intervene in the technology he's using?
I feel like I should pass that over to you. I mean, as you said.
Right, the challenge of, there's been a huge emphasis on genetic disorders, whether they're somatic or germ cell, and because we have ways of dealing and thinking about individual genes and diseases, but we're kind of running out. And with our larger population diseases, it's never a single gene. It's populations or combinations of genes. So I could envision a whole army of these little guys, just like we do multiplex PCR. They just go in there and they fix the genome. So I don't think it has to be one gene at a time, but we need to start thinking populations of genes and key set points. And I think absolutely we could be using CRISPR.
Maybe one comment I'd make there. I think as a therapeutic strategy, I mean, I absolutely agree, many polygenic diseases, the utility of trying to correct multiple mutations, I mean, that is probably not the way to go. But as a research tool, one thing that CRISPR is really starting to revolutionize is, as he said, the ability to multiplex. And so one thing with the ease of designing these guide RNAs to target different DNA sequences, there's been the development of genome-wide screening efforts, where in a population of cells, you can use viral vectors to deliver thousands of different guide RNAs targeting every gene in the genome in one pooled experiment. And if you have some kind of selection strategy, so one way this has been used in cancer is to look for genes that are involved in cancer cell viability, where you basically do a selection, you edit every gene in the genome, and now you see what cells are still living and what cells die, and that tells you something about essential genes for these kinds of cancers to continue growing. So I can imagine, I don't know what kinds of cell culture models there are for epilepsy, but using these kinds of genome-wide screens gives you a way to interrogate multiple genetic influences on certain kinds of phenotypes.
Maybe you could also comment on, particularly with Cas13, the opportunities for diagnostics. So you can start thinking about using these to look at messages and differential expression of mutant versus non-mutant. As we talked yesterday about cell-free DNA and being able to add to that, I think there's opportunities there.
Yeah, so Cas13's a really exciting year for Cas13. It's been developed both for knocking down RNA as kind of something similar to RNA interference or RNAi. It's also been developed into a diagnostics approach for taking blood samples and actually detecting the presence of particular transcripts. I think one of the utilities there, it might be more as a point of care device, a kind of a field test for infection. But it's really imaginative work where you're using this core capability to detect particular nucleic acid sequences. It can be RNA, it can be DNA, and then turn on some signal that can be, report on the presence of that transcript. So I think it, again, well outside of the therapeutic space, there's so many different kinds of applications you can imagine with this core ability to programmably detect particular nucleic acid sequences.
Yeah, and I think you said it, but particularly for point of care of infectious disease in third world countries and things like that, there's tremendous opportunity for early rapid diagnosis of malarias and other things that we can't do easily right now.
Yeah, absolutely.
So conceptually, CRISPR technology can be used to treat or to cure polygenic or multifactorial disorder. But as you know, in a multifactorial disorder, there are multiple genes contributing towards the phenotype, but they are not all contributing to the same degree. So when you are manipulating, you have to keep that in mind. It has to be done in a very nuanced manner so that the end result is exactly the way they contribute. And the other question I have is that with CRISPR technology, as of now, I mean, from what I understand, you can manipulate up to 10 genes, and beyond that, it's very difficult to control, and it could turn into a chaos. There could be a lot of rearrangements, and I see that because I sequence all the time. So can you comment on this?
Yeah, so, I mean, I was just working at a biotech company for the last year, and certainly when you talk about multiplexing, even for 2 different edits you want to make, you're introducing DNA double-strand breaks at 2 different sites in the genome, and those are, of course, hotspots for translocation. So this is something that companies that are thinking about therapeutic strategies involving multiplexing are really concerned about. And even at the regulatory level, I mean, it's the early gene editing trials that I talked about, the kind of things the FDA was looking for in terms of safety were quite crude. It was just, if you take these edited cells and put them in a mouse, do they cause a cancer or a tumor? But now we have the ability to use next-gen sequencing to look in an unbiased fashion at both off-target edits and translocations across the cell population at great depth. So certainly, yeah, multiplexing is not as easy as it might seem. And then the other issue is if you're talking about 10 different sites, yes, you can design the guide RNAs, you can put them into the cells, but you're never going to have 100% editing efficiency at even 1 gene. And now if you talk about 10, imagine the heterogeneity in that population and compounding that with the big barriers to delivering CRISPR into patients. I mean, none of these things are going to be even remotely easy. And I think that's why the companies, the focus is on the most well-studied common monogenic genetic diseases where you have a single edit and there's, complete knowledge about what the mutation is and what the healthy genetic variant is, and there's no questions about what other impacts that edit might have.
So one thing I think that comes up from this discussion and a comment you made, can you do this and can you do epilepsy in a single cell so you can understand this? And the answer, I mean, when you think about the brain and its complexity, not only do you have so many different types of neurons, but you have non-neuronal cells, you have glial cells, you have blood cells, blood vessels. And when you look at these genomic patterns, just even segregating as we've started to do, things happening in different cell populations, how do you deliver this treatment to this cell population and this one to that, because it's not the genes in every cell you want to treat like a tumor. And we're even learning about tumors. The tumors are not as homogeneous as even in a dish. There's chromosomal rearrangements. There's multiple populations of cell types. So yeah, I understand you start with the technology, but we're going to have a lot more technologies beyond CRISPR, I think, ahead to target to specific cell populations at specific times and specific subpopulations of tumor cells. Or you're right, we may have backfiring.
This question is for Dr. Golden. Do you see Brigham and Women's contributing oncology data to public databases such as the Genomic Data Commons, operated in part here at the University of Chicago, where they're bringing together a lot of the data that you spoke of in a public database, publicly accessible database? Or do you see the group staying within its own walls, considering the fact that, if all of the large medical centers were to join together, there'd be so much more data available for all to use? Do you see it one way or another?
Right. So it's a complicated answer, and so let me try and make this as simple as possible. We started with germline, and actually Heidi Rehm and others in our department have created ClinVar, which is a democratized methodology for germline reporting, and everyone can understand it and all the data is accessible. The way tumors are done, the way they're evaluated is different in every center. It's done in different methodologies in each center. And when you try and compare the data, like I say, if you take a tumor DNA and you send it to 3 different places, you're going to get 4 different answers. It's kind of true. some methodologies are really poor at certain regions of the chromosomes, and so you can't rely, and we know that there's parts of our tests that are not good, and having done so many, we know how to interpret it and how not to interpret it. When we send out our data and patients have asked for it to be sent out, we'll give it to somebody else. They look at it, they interpret it differently, and we have to go back and spend a lot of time. No, you can't interpret this area that way. So it's a very complicated thing, and this gets back to something we were talking about briefly yesterday, is that there's not standardization of the data at this time. So we're perfectly happy to give out our data. We'd actually be happy to make it entirely public. We have given it to some data to other places to use as a source of database that are doing the tests the same way we are. The problem is that it creates more problems for patients when they go and take that data and use it in a different system that doesn't understand and know it. So while I'd love to say, yes, we'll make it free, it's great, doing so actually creates some additional problems, actually a lot of problems. And we as a society actually need to figure out how we're going to standardize that, like the germline has actually moved to. The germline's way ahead of cancer, despite the fact that there's a lot more, right now there's a lot more utility in cancer in terms of drugs and things like that. There are some in germline, but more in cancer. That's the problem we have right now.
I would add the problem's even greater than that. A recent publication, can't remember where I read it, but it was recent, comparing from two different institutions, which also had EPIC, and trying to take out the so-called discrete data from both, and then seeing if you can analyze it together, and it can't do it. So we have a problem even at the hospital level.
You know, everyone likes to say, oh, we have EPIC and everyone gets to use EPIC. But the way EPIC is built is that the instance is unique at each site. And so things that get defined by definitions are different at every place. And so even EPIC systems, they can at a very high level, but when you get down into individual data points, they can't talk to each other.
We're out of time?
No, there's 1 minute.
1 minute.
This might be a bigger question then. It was just a follow-up on the standardization. So if there are no rules for standardization,
Stand by. 1 second.
what would it take to, what's the next step? Like, how do we get to standardization? Because it seems like, you know, if that's the log jam, then, you know, let's figure that out. That's not an easy, I know that's not an easy question.
Well, I think it is and it isn't. You know, I think germline has actually done a really good job in that with coming up with standardization. And I think it's going to take someone or some small group to get together and say, and you're probably going to have all the big cancer centers, maybe take all of the NCI designated cancer centers to get together and say, you know what, it's going to be painful for everybody. Everybody's going to have to go through a little bit of a change, but we're going to standardize it. The first step was made by AMP, the Association of Molecular Pathologists, last year, where they standardized the tiering. So we actually used 5 tiers. We have to go to 4 tiers. Everybody's going to be, well, the recommended standard is now going to be 4 tiers of data, which are used everything from here's actionable down to this is a variant of unknown significance and or is one of known not to be pathogenic. And so now I think most places have agreed to go to this 4-tiered system. That's a step in the right direction, but it doesn't address the actual collection of the data and the data itself. And so that's the next step that we have to go to, and I think it's going to require a body to get together and do that.
I would argue there's potential danger in standardization. I know in everybody's field in medicine, you meet every 10 years and you reclassify and you sit there and argue vehemently and everybody's heated, you know, this is the better classification than this. If trying to spend all the time bringing everything together may be a rather large challenge, but then it also limits that everybody has to do it this way, but if one lone institution is doing things in a unique way or storing or collecting data differently, that could be the next CRISPR, or that could be the next discovery. So what I think can be done is how data can be linked. So one institution may do it one way and another. All you'd have to do is say, how do we create the links? And then you tie those bits of data. So you don't have to transform the data as the genome changes, as names of genes come up. We find ways of creating those linkages to actually translate between those two areas. So there's standardization and trying to make everything rigid, and then there's ways of creating linkages, I think, with existing or novel data sets as they come up, and just keep adding more and more into it. And I think that, to me, is more exciting.
Okay. I think we're out of time, sorry. So I want to thank both Jeffs and Sam for very exciting talks and information. So